In this video, we will look at how we can tag and label proteins. So, why would we want to tag or label a protein? Well, in the lab, we often want to produce proteins for experiments, or we may want to produce a protein for the treatment of a disease or condition. However, the cells we use to produce the proteins will also be making the proteins they need to live. And this means that our protein will be mixed in with all of theirs. So we need a way to purify it. And that is where the tag or label comes in. It gives us a handle by which we can purify our protein. Another problem we face is making enough of the protein to purify. And to overcome this problem, we must grow a lot of the cells. However, there is a problem. If we are growing the cells to increase their number, then as they are growing and dividing, the making of the protein we wish to express will be a burden and will slow cell division and hence protein production. So how can we control protein expression and also purify the protein? To control protein expression, we can use an expression vector with a component that regulates protein production. The regulatory element that is often used in bacteria is based around the lac Operon mechanism and uses a chemical called IPTG to relieve the inhibition placed on the protein production by the vector. So, once you have grown the bacteria to the required number, you can then add IPTG to turn on the production of the protein. So, we now have the cells making a lot of our protein, but how do we purify it? Well, there are two approaches that can be used. In one approach, we engineer the protein to contain an amino acid sequence that targets the protein to the space between the bacteria's inner and outer membranes, as this compartment contains very few proteins, and so our expressed protein will be in excess. In the other approach, we tag the protein either with a hist tag, which is just a series of 6 or more histidines on the NLC terminal, or we add a protein called GST, which stands for glutathione S-transferase, to the N terminal. In both cases, the tag is added in the expression plasmid as the DNA at the start or end of the DNA we wish to express. After the cells are lysed, our tagged proteins are purified using affinity chromatography. That is, beads coupled to glutathione for GST tagged proteins and nickel for the hist tagged proteins. One advantage of using GST is that the proteins can be released from the beads by using an enzyme that cuts between the protein we are trying to produce and the GST. One final type of protein tag that I would like to mention is GFP, green fluorescent protein labeling. GFP is a fluorescent protein from jellyfish and is used in the lab to label proteins so we can track a protein's movement through a cell using a microscope. And while GFP labeled proteins have nothing to do with protein production and purification, it is worth mentioning here as it is a technique that, like GST and hist tag proteins, relies on the cloning of a marker and the protein of interest into a plasmid so both are expressed as a single protein molecule. Okay, so that covers all I want to say on labelling.